for this guest lecture. I hope that this lecture will be very useful to you because it's a very important problem which we are facing for several decades. This Begomovirus infestation in pulses. It is a menace in the pulses cultivation. So I think Dr. Reddy's lecture will enlighten you what are all the molecular methods which we can adopt to identify different strains of this Begomovirus which are prevailing, especially in Andhra Pradesh. We are having some adjoining areas to Andhra Pradesh state also. So where we can also get similar kind of infestation. So this lecture may be useful for the students, those who are working, especially in case of Begomoviruses. Dr. Reddy, please.
Good afternoon to all. First of all, I should thank the TNAU University authorities for inviting me here as an examiner, and as well as, first of all, I should thank my classmates and professor and head plant pathology, Dr. Angappan, and for giving me this opportunity because I came here. It's the third time to visit TNAU and other staff members. Actually, initially I worked in groundnut for 5 6 years on groundnut bud necrosis and stem necrosis disease. It's a very important disease. But in 2010, because in Andhra, the so-called famous varieties like LBG 623, 645, they are the ruling varieties. LBG 752 also. They are ruling varieties, not only in Andhra, throughout in many areas in the country also. The popular black gram varieties. But in 2010, they became susceptible to this yellow mosaic, and you know very well, it's a very dangerous disease. The losses will be 100% sometimes, particularly if you go for late Rabi, 100% in Andhra. Of course, Kharif, the incidence may range from 50 to 70%, sometimes 20 30% also Kharif, but big problem in late Rabi or in summer crop. So then there was no variety to recommend for cultivation, and the farmers, if you no farmers shifted from black gram to maize, Guntur, Krishna, Godavari, because they grow a lot of pulses in rice fallow system. I think rice fallow system is also there in Tamil Nadu because before harvesting rice, what they do is they broadcast the pulse black gram and they harvest the rice and they leave it on residual moisture. That crop will come within 75 80 days. So that was called rice fallows. And they have no option, no variety to withstand this YMV. So they went to maize. And government asked what to do. Then they recommended PU31, one of the famous variety from Pantanagar, PU31, as a source of is a highly resistant to YMV. And the problem with that PU31 is no, it's a very small stature. So YMV resistant, small stature, and rice fallow system, it has to dominate the weeds. So after harvesting the rice, they don't bother about the weeds and other things. So crops which can grow taller or medium size, they can survive in the weeds. Some people go for weed clean cultivation and all. Otherwise, so that was situation, and PU31 came into the picture. And one more problem with in Andhra farmers is Andhra farmers prefer a shiny black gram variety, shiny, glossy, shiny black gram seeds. Otherwise, I think in Tamil Nadu and all, they use normal black gram in North India also. But our farmers, they prefer shiny, and which these varieties give more income also, maybe 200 to 500 rupees per quintal, extra income also. So we don't, due to lack of shiny varieties and lack of resistant source, suitable varieties for Andhra, except so there was a pressure from the government as well as university to solve this problem. And then I shifted from oil seeds to black gram initially. So then that is how we started working from 2010 onwards. Right from I collaborate actively with my principal scientist, the Pulses, who is now director of research, Dr. Prashant Madam. We work together, and right from marker development, virology one part, markers development one part, and how to develop a variety. So this is actually a lot of work has been done in this university. I know on Begomoviruses, And this is a leading center in South India for Begomoviruses, particularly on pulses, mung bean, and black gram also. And whatever I have done, uh, of course, it includes my student work, one or two student work also there. I will show some light on that. So this is a typical symptoms. I think most of you know the YM symptoms on pods, and you know very well it is a Gemini virus. It is transmitted by whitefly, circulative manner, and all of you know it's a bipartite virus. It has a DNA A component and DNA B component. The size is 2.7 kb, and DNA codes for code protein and replication function, and the DNA B codes for movement protein function. And A and B are required for infection. So far, I don't know anybody has got the infection with DNA A alone or DNA B alone. So you have to mix DNA A and DNA B in this case. I think in tomato you can get partial symptoms with the DNA A tomato. If you mix with DNA B, you get more symptoms. But here in pulses, so far, if we inoculate DNA A, we don't get symptoms. If you inoculate B, we don't get symptoms. If you mix A and B only, we can get the symptoms. And A is more or less similar. If you see the sequence homology studies, they don't differ much from one district to another district. But B is highly variable. A good work has been done in this TNIU where they have isolated 5 variable Bs from one black gram, I think Mungbin, Dr. Usharan, I think, where one B, the varieties which are resistant to 4 DNA B variants, they are susceptible to fifth one. So that is how this DNA B is highly variable. And in legumes in India is mostly infected by 4 species of Begomoviruses and Mungbean yellow mosaic India virus, Mungbean yellow mosaic virus, Asgram yellow mosaic virus, Dolichos yellow mosaic virus. And the literature says up to 2014 in Tamil Nadu, mainly in South, MYM is responsible for YMD disease. And what we have come in our state in 2014 that both are there, both the Mungbean yellow mosaic virus and Mungbean yellow mosaic India virus in 2014. Now that is true now throughout India also, even Tamil Nadu also, even North India also. They have reported both the species of Begomoviruses, both Mungbean yellow mosaic India virus and Mungbean yellow mosaic virus in black ground. I think that is true with Mungbean also. So that paper came in 2015. And these are the common primers everybody use in detection of Begomoviruses. Some of these are the common primers. You can detect a full length genome or a partial code protein. Some with this design. So these are the common available in the literature. So when in 2014, when we did we amplified samples from 4 districts, Guntur and Vijayanagaram, Tirupati and Kurnool. We sequenced this code protein gene sequence based on code protein sequence homology. We came to know that Mungbean yellow mosaic India virus. Up to that, earlier paper says that Mungbean yellow mosaic virus is with pulses, particularly black gram. We reported that Mungbean yellow mosaic India virus is there. Of course, now that is a true. Now a lot of publications came afterwards. This was the, and during that time we did collected some more isolates from Rayalaseema, Telangana, as well as coastal area, and coastal they are forming a separate the variant, maybe Rayalaseema and Telangana, maybe another. See here, Begomoviruses they keep changing. It's not universal. They don't fix also. Even if you take isolate from maybe isolate from this year and compare with next year or next season isolate, there must be some differences. So, and this is the picture. One of my students, Dr. Chaitanya, she is now scientist at Nandyal in chickpea program. She tested based on PCR detection only, not based on sequencing. Different pulse crops she took and she tested by PCR this, and she could detect both the species in so many crops like black gram, green gram, horse gram, pigeon pea, cow pea, French bean, field bean, soybean. So you can get both the species of Begomoviruses, and we can detect some satellites also. Of samples see tested. Uh, okay, so both the species are there. Of course, the sample size is very small, but still it gives very good information that both the species are infecting pulses in Andhra Pradesh. So even if you see the month wise also, of course, the sample size is very small, but the MIM is more prevalent in September, October, November months as compared to MIV, that India virus, in December, January, February, March. Of course, you need more samples to say this, but why this is important is one of our variety, leading variety in Andhra, LRG 752, is 752. Even I think it is there in other parts of country also, which is resistant at few places, but they are susceptible to some other places. Like in East Godavari, Krishna, and you go to Guntur, some places farmers say we don't get disease on 752. In some places we have seen ourselves in Goa near Pattisima and other places. There is a lot of disease. Later, through agroinoculation technique helped us to find out that the 752 is resistant to one species but susceptible to another species. So there is a differential reaction. So really this agroinoculation technique helped us to find out why we are getting a virus on in particular area, infection particular area, but not in other areas within the district or entire state. So this information helped us. Similarly, we have tested, these are different district samples. Of course, even under ICRP, ASRP, I got some samples from Tamil Nadu also. This is 5-6 years back. This I presented in SK Nagar. I think anybody came from to SK Nagar or ASRP meet, they know. In TNAU that time, I think it's 7 years back, 5-6 years back. So both are there in Pantanagar also. Similarly, Akola is only one. We got some samples from Varisa also. I think there is another table is there. Yeah, here. So you can see in, of course, this is a based on PCR detection only. So in Pantanagar also we got the both in Coimbatore, in some other places also. So now the picture is latest literature says that both the viruses species are there. Most of the most part of the and sequence similarity matrix just I calculated for our own isolates. Since from if you see since from beginning 2015 first Tirupati isolate, then 2022 in black gram, green gram 2022. If you see 7 years, not much difference in the DNA. As I told you, it's more or less similar. Not much variation will you will find in DNA. Of course, we have to go for more isolate sequencing to say this word, but whatever we have done, not much variation in DNA. For this is the phylogenetic tree. Mungvinala mosaic virus India A. Here also we have first isolate in Tirupati 2012. We sequenced the full length in 2012, then 2015, 2020, even 22 also. So there is some variation up to 99, 94, 93, 93. Particularly Mungvinala mosaic India virus. One thing which I cannot understand is okay, the variation is always there. Any virus we have variation. You take isolate from Coimbatore and a nearby station. When you do, when you will find some sequence dissimilarity or similarity will be there. Whether that is going to play any role in symptomatology, that we have to see. That is very important for us. This is the same tree generated. So B, yes, we can find a significant variation in the B. Particular, what we observed is first, especially with East Godavari isolate. B with East Godavari isolate, we found some variability, variation. We wanted to see whether that variation has any impact on the symptomatology or breakdown of the disease resistance. We made some agroinoculation constructs. We came to know that yes, it has some disease develops early than as compared to other isolates. It's the same thing. Yeah. So you can see here, where I have some other photos. So this is East Godavari. Here, this is comes 1 week before the other Prakasam